Hello, everyone, and thanks again for joining. My name is Christian Yannis Kuzanetti. I'm the CTO and one of the co-founders of Fluxion, and it's my pleasure to introduce this talk. This talk is aimed at BioFlux applications relating to biofilm research, and biofilms have been one of the first applications that were developed for the BioFlux. It's been a great area for us to work in, and now there are a number of people that have published great contributions to this field. And Brian Haynes, who's our Global Director of Sales and Service for the BioFlux, will be introducing a few of these key applications. Thank you.
Thanks, Christian, and thank you all for attending today's talk. Just like the early pioneers of biofilm studies were done with oral microbiologists, early adaptation of the BioFlux system was largely performed with oral biofilm researchers. Today I'm going to talk briefly and give an introduction to oral biofilms, talk about previous methods for growing biofilms, and explain how the BioFlux system works. We'll then review studies using BioFlux to better understand oral biofilm biology. Talk about agents to disrupt biofilm and the role of shear on microbial adhesion. This was just in the local paper in August of this year. The Jefferson Memorial is on the National Mall and is covered in biofilm. I can only imagine how many biofilms the paper publishes every year. I doubt it's that many. It must have been exciting for this reporter, exciting knowing something he learned in biology turned out to be relevant. One of the central themes of this article is that the National Park Service is looking for agents to disrupt the biofilm rather than totally destroy it. It's fairly easy to blast it off with bleach and a power spray, but that would likely hurt the marble and damage the rest of the environment. We're going to return to this concept of disruption in the papers we'll review today, the idea of not necessarily eradicating all biofilm, but instead disrupting it and working with it. I live in a small town and really love to talk about my work when I can. And people often get confused when I talk about biofilms in the context of disease and the environment. And the easiest answer I can explain is the cheese. McDonald's French fries are essentially planktonic bacteria, where the cheesy fry from Outback is a biofilm. Like antibiotics or macrophages to biofilm, you can't eat through the cheesy fry. It's just not possible. You can try, and it'll certainly fill you up, but usually one person alone cannot tackle the cheesy fry from Outback. And that theme goes along with many antibiotics and mechanisms of the inflammatory system have trouble dealing with the complex structures of biofilm and being able to both penetrate its outer layer and to tackle this problem. The International Union of Pure and Applied Chemistry defines the biofilm as an aggregate of microorganisms that are frequently embedded in a matrix. I like to think of a matrix of cheese, to adhere to each other and to a surface. It's easy to see how the oral microbiologists were able to describe biofilms so early. It was literally staring them in the face. And one more example, here we have a distinguished gentleman from the British Intelligence Service, and he has an accumulation of biofilm. This picture is a little fuzzy, but it's along the gum line there and probably some periodontal issues as well. Back in 1932, it was published that it is quite evident that for the most part, water bacteria are not free-floating microorganisms, but grow upon submerged surfaces. This is a favorite historical paper that biofilm researchers like to quote and put on their website, like George O'Toole. I think it's rather poetic that Henrique was essentially describing BioFlux research in the 30s. In terms of research money currently granted by the NIH, there's about $200 million per year being spent on biofilm research. And honestly, it's not enough. Given that around 80% of infectious diseases have a biofilm component, the biofilm researchers should be getting a larger chunk of this $5 billion per year being spent. I would encourage you, if you live in the United States, to write your senator and congressional representative to support biofilm studies and keep the NIH funded. One of the interesting things about oral biofilm research is the number of different microbes growing in the mouth. It's estimated that at least 500 different species are there. In Alex Ricard's lab at the University of Michigan, he sequenced the composition of oral biofilm growing in the BioFlux using saliva. A lot of these are expected, like Streptococcus. But the one that really is interesting is this TM7. There isn't much available in TM7, so I took the liberty of looking it up on Wikipedia. Between Wikipedia and Facebook, this is where Donald Trump gets most of his foreign policy guidance, so I figured it wouldn't be too bad to look at. And, you know, just as a general, TM7 is one of those viable but not cultural bugs that they talked about in microbiology, saying that, you know, the things that we wind up growing on LB slabs, that there's a lot that's in the environment that just can't be grown that way, even though you see lots of fuzzy stuff on your LB slab. You know, it's largely found through 16S sequences in odd places like sponges, cockroaches, and mouse. I'm going to describe some of the previous methods for studying biofilms. Each has their place and are useful, but there are certain limitations with each one of these. Crystal violet is very popular, and it's rather inexpensive. The basic procedure for this is to inoculate a culture in a plate and grow it to a stationary phase. You add crystal violet, incubate it, you pipette out the planktonic bacteria or the McDonald's French fries, and then you solubilize it in ethanol, and you measure it at OD 600. You know, when you compare that with a biofilm experiment using BioFlux, the basic procedure is that you would prime the plate, inoculate the culture, fill with source media, set your stage positions, and do a time lapse every 20 minutes for several hours. You know, if you were going to copy that, you know, data point by data point, you would need to use 60 plates because there's 60 time points in this description here. You don't have the option to monitor the same biofilm over time when you're using crystal violet. There's a limitation to the amount of fresh media and certainly the amount of shear that goes onto it. It's not possible to really image different strains. For example, if you had you know, one strain that was expressing GFP and another strain that was expressing something like cherry or tomato, you wouldn't be able to see that in the crystal violet staining. Likewise, you can't really tell what's alive and what's dead. What you can tell is the material that's stuck to the surface and what hasn't, what's stained with crystal violet, and so the readout is the material. The real limitation I find with this is that you need to pipette it out, and that I find, you know, when I'm pipetting, depending on the time of day that I'm pipetting, it could be a little different just with myself, and then, you know, from researcher to researcher, that variability increases. The adhesive property of the biofilm is dependent on how aggressively you pipette, and if you notice here in this well, I mean, that's right where the pipette was going, and that's what blew off the biofilm. This is a previous paper that came out in 2011 using crystal violet, And the point got across in terms of what they were trying to make about this drug that they were using. But there's a lot of heterogeneity in these samples, and you can see differences in how this material stained in the wells. It's not possible to see if there's live or dead, and the hammer dose is really where it shows the most effect on this paper. And actually, if you look at these wells, the sort of ring of crystal violet, I kind of question whether or not there's actually material there, or that's just really an artifact of the assay. So my wife has a PhD in biology, and when I was going over a talk back in January, the term flow cell kept coming up, and my wife kept asking me, what cell in the body are you talking about? And so here it is. A flow cell is consisting of an inlet, an area of perfusion, an observable window, and an outlet, most commonly used for live cell imaging. Here you can see a couple cartoon and real pictures of flow cells. Most use several liters of media and have a limited number of actual cells. The number of actual cells that can be imaged in real time is even more limited. So on this bottom left panel, you see that these slides have three, and you can't really see it from here, but they have typically 3 individual flow cells on them, and they can sort of daisy chain this to make it so that they can have 9 flow cells running, a lot of media and stuff like that. But if you were going to try to image this in real time, you'd only be able to put one of these slides on the microscope anyway. One of the really interesting devices that's been used for studying biofilms has been the Robbins Device. It's reusable. You can see the yellow plastic, or how yellow the plastic is, because it's autoclaved after each use. And coupons allow for large amounts of biofilm to be removed. It's limited in the amount of volume needed. Even heating is difficult because to be able to heat the whole system evenly. And you can see in this room is like a 37-degree room that's being used to evenly heat the entire system. And only really one condition can be processed at a time. Coupons were a little more relevant back several years ago, but with technologies like next-gen sequencing, sensitive Western blots, mass spec, it becomes less important to get massive amounts of biomaterial for your assays. The CDC flow cell typically has 4 channels. Each requires large volumes of media and cannot be imaged in real time. It also requires a lot of assembly each time it's used. And I have here on the bottom right the number of different parts that you have to put together, autoclave, and then take apart, clean. And, you likely have to autoclave these before and after. I once heard a researcher refer to these systems as supermodels, each one with their own strengths and weaknesses. I think Trump would probably say this is a 4, and Hillary would simply say that all flow cells are beautiful. This holds 4 individual slides, and in order to operate it, you need to coat each slide before starting, and then clean off each slide in order to image it. The CDC reactor is a classic tool for biofilm use. These are great systems. However, they require huge amounts of media. They operate off of, I call them dipsticks, but basically sticks that go into the reactor. And it's been reported by many different researchers that just by pulling these sticks out, no matter if you pulled them out at an angle or you pulled them out slowly or quickly, that just the surface tension of the media would wipe the biofilm off of the coupon. This entire system provides really one condition of media, and compare that with other systems like BioFlux, where you might have 24 independent flow cells. This is a an image of a CVC flow reactor from a customer site. This was done in a 37-degree room. And you can see the amount of media required, that the number of peristaltic pumps. And they do have coupons, and there's certainly something growing here. They're really only able to see, one condition at a time. Sorry, it showed that my screen had disappeared, so I wanted to make sure we were still online. It looks like we are. The BioFlux system is an array of flow cells. So we established, what the definition of a flow cell was. It operates using microfluidics, where liquid is perfused at controlled shear stress using air pressure. The flow cells are part of an SBS-type plate that has a glass cover slip bottom. So if you imagine the standard type of tissue culture plate that would have either 48 or 24 or 6 wells, that's the type of plate that's used as for the inlets and the outlets. So just to briefly, explain this, the pneumatic air pressure seals onto the plate and pushes liquid from the inlet to the outlet, or the outlet to the inlet, and a microscope objective comes from underneath. This is looking at the channel characteristics of a BioFlux plate from the bottom up, where one channel serves as an inlet and one serves as an outlet. Here you can see 2 independent flow cells in a 48-well plate. On the bottom right, there's a diagram of the 24-well plate that has 2 inlets and 1 outlet. In the 48-well plate, you have 24 individual flow cells. We have the option called the Quattro System, where 4 plates can be operated with 1 controller, and in that case, you can have 96 flow cells at one time. This is where the viewing channel of a BioFlux plate is, and where you would image the biofilm growing. And on the right, you have a 3D reconstruction of what an oral biofilm would look like growing in the channel itself. One of the critical differences between the BioFlux and other systems is that liquid does not touch the tubing. The only liquid touches the consumable of the instrument. All fluids are kept in the consumable. In most other systems, the liquid perfuses through all of the tubing. Temperature control of the tubing is nearly impossible unless you put the entire apparatus, including your inlet vat of media, your tubing, your microscope, and your outlet, all in a 37-degree room. And just to point out with this device here, that you see these individual tubes. Each one of these tubes needs to be primed and then attached on before operating the machine. And that actually goes both ways, both on the inlets and the outlets, in order to operate this for any type of extended period. With the BioFlux, this priming is all done actually in the plate, and there are no really tubes to attach where liquid might be, sort of dropping or dripping out. This is a time-lapse image of an oral biofilm being grown. Earlier, you probably saw a cheek cell in the channel, though you can see it still right now. This is grown in LB. Growing culture in cell-free saliva would have produced a slower, more stable structure. But you can see as the biofilm accumulates and then sort of disperses back. This was my cheek cell. I'm not sure if I made that clear earlier. But probably the question you've all been wondering about, this is my dentist. If you want her number, I think she's taking new patients. She's got very nice teeth, too. That previous video that you saw was really, it helped fit the model of what this classic cartoon is for a biofilm growth, where, very simply, you have attachment, growth, and dispersion. I mean, lots of technical terms in there, but that's the gist of it. Now, depending on the organism that you're growing, some are more likely to have this dispersion phenotype versus others. And depending on the amount of shear going on, some are more likely to roll versus others. Some of that can be modified simply by the coating that you put on the channel. For example, if you coat the surface with a particular protein that might help the bacteria adhere longer and harder, that might help model what you're looking for if you want to keep all of the biofilm on the plate at one time. In the case of Dr. Ricard's research, he coats the channels in saliva, which preconditions the channels with protein and polysaccharides and everything else that's in saliva to make sure that the bacteria have something very happy to grab on. In the image that I showed you, this was basically directly grown on glass, minus whatever proteins came out of LB before I inoculated the culture. This is an example of a side-by-side oral biofilm growth. The top is my mouth untreated, and the bottom is treated with a fraction of toothpaste from a cosmetic company in Korea. So I took out the abrasives and spun it down, diluted it, and still you see a rather remarkable attenuation of the amount of biofilm being formed here. When you go to give talks to, for example, investors or other groups of people who want a quick visual representation, this type of side-by-side video is very useful. This paper helps set the stage for a lot of Dr. Ricard's work at the University of Michigan. A central theme of most of the biofilm studies coming out of his lab are that they use cell-free saliva instead of artificial media to grow and study biofilms. In addition, as I mentioned earlier, the channels are pre-coated in saliva to condition the channels for a nice, suitable matrix that the cells can sit down on. This is a live dead stain of oral biofilms, and you'll see a lot of images that look like this throughout the talk. In general, red is dead and green is alive. These stains are done after the biofilms have been established. Cyto9 stains DNA of viable cells, where PI stains DNA of dead cells with a disrupted membrane. One of the limitations to using PI is that it stains extracellular DNA. Cytopyridium chloride, or CPC, is a ammonium compound used in types of mouthwashes, toothpaste, lozenges, throat sprays, and whatever else Wikipedia says it is. So going through this panel here, you see control, PBS, and we expect that would all be alive, and 70% ethanol, we'd expect that would all be dead. And then this is starting at 0.5% CPC and going down to 0.001% CPC. And the majority of mouthwashes that contain CPC are between 0.1% and 0.05%. So that therapeutic index you see here, where it's basically killing off all the biofilms, is consistent with where mouthwashes are today. This is the second paper in a series from Dr. Ricard studying the role of arginine in the disruption of biofilms. One more note about the medium of the biofilms being grown in the lab is that all donors had not taken antibiotics for the last 3 months and didn't smoke. The saliva from each donor was pulled, DTT was added, it was centrifuged down, filtered, and then diluted in distilled water, as pure saliva is a little viscous. So this is saliva, but it is slightly diluted, and the protein is just slightly denatured. But otherwise, this is a primary medium compared with, say, using LB, like I was using in those earlier videos. This is the volume of biofilm being grown in increasing concentrations of L-arginine. And you can see how the quantity significantly decreases over time. And actually, this theme carries out through a couple of the publications that Dr. Ricard has done, where actually no amount of arginine shows slightly less growth than something like 5 millimolar of arginine. And then, of course, as you get to this 500 millimolar or 250 millimolar range, that the growth is significantly less, but you don't see a lot of red. It's more like it's disrupted in this case. This slide is looking at the operational taxomic units of the biofilm grown in regular saliva and saliva treated with large amounts of L-arginine, in this case 500 millimole of L-arginine. Again, TM7 is very interesting in this situation where you see that after the treatment with L-arginine, the TM7 fraction actually increases quite a bit. And I have to wonder if L-arginine increases TM7 in oral biofilms, kind of wonder what it's doing in cockroaches. But anyhow, that's for a later time. When you look at the biovolume of different species, you see that L-arginine completely changes the abundance of the bugs in the mouth. So not only is there less biovolume, but the proportion of the species that are currently there is totally different in the L-arginine case versus the case with just saliva alone. And in particular, what's interesting is that the amount of Neisseria is lower in the L-arginine case. These are images of live dead staining again. Control water, 0.05% CPC, which we know from the previous paper completely kills off most of the biofilm, and a high dose of the CPC, the 0.05%, which is used in mouthwash today, and L-arginine. This is where it gets really interesting, that where you see that 0.01% CPC does not fully kill off the biofilm, and 500 millimole of L-arginine doesn't kill it, but does seem to disrupt it. However, the combination of 0.01% CPC and 500 millimole of L-arginine both disrupts and kills off the biofilm. And you ask, well, how is that really significant? And one of the rationales is that CPC is not necessarily tolerated by all patients that would wash their mouth with it, and that by decreasing it, fivefold, that perhaps this is something that's more tolerable to be able to rinse out with compared to 0.05% or 0.1%, which some people are just simply sensitive to it or don't want to rinse their mouth with it every day. The third paper in this series, arginine is studied in the role of biofilm growth. What you'll see is that there's an optimal level of arginine required for biofilm growth, and too little or too much is essentially disruptive. This was looking at Streptococcus guerondi, I'm hoping I'm pronouncing that right, which is a little bit different than just using a mixed culture of oral bacteria. It looks largely like only Cyto9 was added to the biofilm, but it's possible that nothing was dead in these samples. As you can see between the as you can see between this side and the next, there's an optimal middle ground for L-arginine concentration that very high levels are disruptive to growth. The other thing to consider is that Strep guerondi, it just simply has a different phenotype than the mixed species. So when you see this looking a little different than the previous papers from Dr. McCarty, just keep in mind it's a monospecies in this case. And they graphed out this data here and it shows whenever the control all the way to 500 millimolar of L-arginine, and of course the 5 micromolar range. Actually, here it shows more than 50. In the other paper, the 5 micromolar seem to have the nicest sort of biofilm, biovolume thickness of it. And the conclusion of this story is a happy one in my mind. This patent application just published, and I'm really excited to include it in my talk today. Publications using BioFlux are exciting, but patents using BioFlux lend to the significance of the instrument. There are several patents and patent applications in both platelet biology and biofilm research. that use data from BioFlux in their images and that helped enable their claims and reduce their invention to practice. As this theme continues in the last paper we reviewed, you see where they describe it and illustrate out that there's a balance between the right concentration of arginine to biofilm growth. You know, I counted, just because it was exciting to me, that 8 out of 12 images in this patent have images of biofilms from BioFlux channels in it, which I think is quite exciting to me. And it's definitely validation for the technology being useful for downstream research purposes and the protection of research. There are 2 more papers that I want to review on the topic of anti-oral biofilm research. These are both from the 4th Military Medical University in Xi'an. This is one of the most prestigious dental universities in China. This first paper was looking at properties of BAC8C. It's a custom synthesized compound largely studied by this group. This paper looks at strep mutans and grows them in BHI media. They use chlorhexidine as a positive control. Some of you may have used something called Paradex after a deep cleaning at the dentist's office. It's really nasty stuff. I've had deep cleaning several times and just stopped getting the prescription filled because I never use it. It's interesting, at the lower concentration of BAC8C killed off more than the Paradex control that was 10 times the concentration. You see, you have the control here and then your Paradex, or CHX as they call it. And then really at the highest concentration, you see that it's about 10 times less than the BAC8C, and that, according to the quantification mechanism they were using with the software, that killed off more of the bacteria than this control did. Not really sure, however, if BAC8C tastes any better than Paradex. I guess that will be determined at a later time. Pleurocidin is produced by, I'm probably pronouncing this wrong, Pleuronectes americanus. It's an antimicrobial compound. I never took Latin in high school or college, so I'm sure my pronunciations are quite terrible. This study also used strep mutans and BHI in media. As many of you know, strep mutans are the first colonizers to the teeth after you totally clean them off. They like to grab on first, and so it's understandable that they would use strep mutans in this study. Here you can see the effect of pleurocidin with controls of Paradex and 70% isopropanol. I want to point out, however, that totally eradicating biofilm may not be necessary. We understand that there will always be bacteria in our mouth, and that having the right balance of microbes may be a sustainable answer to oral health. And getting back to the Jefferson Memorial, I mean, we really can't just tent the entire thing and spray in ethylene oxide and hope to totally sterilize the entire, and of course, even if we sterilize it, the biofilm will still be there. It's just will be dead. That I hate to put it in such a liberal way, but getting along with biofilms might be something that we're headed towards instead of completely wiping them off the face of the planet. Just so it shows the effect of pleurocidin on an established biofilm. In panel A, you can see the inner core of the biofilm is being protected. Again, red is dead and green to live, where it's thick enough. The outer surface of the biofilm structure are clearly more susceptible to the drug, and it appears that the matrix and the surface of this biofilm helps protect at least the inner core of it and keeps a portion of it alive. This paper came from NIH out of Rob Palmer's lab. Dr. Palmer is one of the pioneers of oral biofilm research and an early adapter of the BioFlux system. Many people have cycled through his lab and have gone on to start their own labs in academia and in industry. He really was a pioneer of the field. I believe he still works at NIH. I'm not sure how active his lab is right now. I'm just going to show one figure in the interest of time. This is looking at strep gordoni and BioFlux channels coated in different concentrations of pituin, a blood protein or saliva. DL1 is the wild-type strain of the strep, and D102 is the HSA deficient mutant. HSA protein is involved in cell adhesion. The left panel is the number of cells bound to shear forces, bound at different shear forces. In this case, they use Pascal. 1 Pascal is equivalent to 10 dynes. And you can see the HAS mutant did not adhere well at all. Interestingly, the lower dose of pituin had a better adhesion than the higher dose of pituin. Actually, I take it back. Now that I read this again, the higher dose of pituin had it. So I was wrong on that one. I need to correct my notes here. The higher dose of pituin had the greatest adhesion. The saliva actually was somewhere in between the 25 micrograms and the 5 micrograms. The right panel shows the fraction of cells that remain stationary rather than rolling on the surface of the channel. In this case, wild-type perfused over saliva-treated wells had the most stationary adherent cells. So to conclude this talk, in the interest of not going over a little more than 35 minutes, this image shows, primary media can be used to study oral biofilms. And that's significant when you want to be able to do more than just one reaction at a time. If you were to use those large vats in the traditional flow cells, you'd have to get half the campus of a particular university to be spitting into it in order to get enough media. In the case with the BioFlux, 2 donors can do an entire plate with saliva. Mechanisms and compounds can be characterized using BioFlux. Oftentimes I ask researchers, are you looking at mechanisms or are you looking at drugs? And sometimes it's both, but usually it's either looking at specific genetic mechanisms, looking at drugs, or looking at the genetic mechanisms, finding the drugs that affect those, and then characterizing them. And in general, BioFlux enables flow-based biofilm growth with higher throughput and smaller volumes of media and compounds. I want to thank you all for listening today. To my friends on the West Coast, thanks for getting up so early. To my friends in Europe, it's time for beer. And for my friends in Asia, this was all a dream, and you go back to sleep. Please feel free to write any questions in the panel.
All right, everyone. This is Christian, and I just wanted to relay some of the questions to Brian so you can get to them. One of the questions was, any thoughts on how some of the flow systems have the main advantage of giving you access to the biofilms to test, or do viable counts? And
well, this was done by Christian. There's a little bit of an echo. Can you mute your phone on your side, please?
Sure.
So this was done in Dr. Ricard's lab in Michigan, where, and this is described in his paper, but very simply, to push the biofilm out of the channel, he pulsed it with 20 dynes, and that popped it out very quickly. In terms of viability, the easiest way to look at viability is with the backflight kit. If you want to look at, CFUs is sort of an interesting challenge in the context of a biofilm, but if you were to push out the biofilm, somehow disrupt the biofilm and get it into a single cell phase without killing it, and then plate it out and count the number of CFUs, you could do that. Or if you weren't content with propidium iodide and cyto9 or the other markers for viability, you could take the amount of material and, for example, put it on a cell respiration machine and look at the amount of respiratory capacity out of it. You could take it and put it through a meter that would look at electrical conductance and determine whether or not that's alive. Let me go down to the rest of these questions here. Does the quantity of biofilm produced with the BioFlux allow to study the transcriptome? And yes, it does. In subsequent papers by Dr. Ricard, he's looked at next generation sequencing. I'm not sure if all of them have been published at this point. The first one was, of course, with 454. However, you can just as easily put this on a system from like iron torrent or aluminum. There's definitely enough material there to look at the transcriptome or to amplify and then look at the transcriptome. Next question is it possible to recover the material? Yes, it is possible to recover the material. Metabolites, for instance. Yes, of course, it does depend on your limits of detection, but there's more than enough material here to look at, say, respiratory capacity. And I had one more question about using the Quattro system, how many samples that they had missed part of the talk. So 96 flow cells can be operated at one time using the Quattro system. You could image one of those plates in real time if you wanted to, or theoretically image them all at the end. A lot of these researchers run their samples and then stain them and image them at the end. I have a preference to do both ways. I like to do real-time imaging to see what's happening, and then at the same time, I like to have the option of doing further staining or downstream analysis. And with the system, it allows you to, for example, do the imaging in real time and then take that exact same biofilm and then do your downstream analysis with it.
There's one more question, Brian, which is how long can you grow an established biofilm for using the system under flow conditions?
Oh, thank you. In the 48 well plate, and this of course depends on how fast you want to run the biofilm, but you have close to 70 hours in a 48 well plate that has 1 milliliter of media running at 0.15 dyne. With a 24 well plate, you have 3.5 times more media per inlet well, and so you principally have about 3.5 times that. And of course, that also depends on how fast you want to run it. We came up with a 6 well plate that allowed for extended flow, and the goal behind that was for these really longer term studies where you're looking at something like tuberculosis or a long-term infection where the population, which may be mixed in culture, starts to evolve and change over weeks. And with these with these 6 well plates, we have close to about 3 weeks that you could run this without actually having to pause and exchange the media. If you're willing to pause and exchange the media, you can go for as long as you'd like to. But the idea with the 6 well plate is that you can set it and leave it for extended periods of time without having to actually open it up. Christian, did you see anything else I missed?
I think that's it for the questions. If you have any other questions that come up.
One more.
Okay, go ahead, Brian.
So it was asking about an anaerobic environment maintained either inside or outside of a glove box. The BioFlux is defaulted to use room air as the air pressure that it pushes the liquid from. But that can be bypassed with an environmental control option where a custom gas mixture can be pumped in. Now, the gas has to be pre-mixed before it goes into the system. But something like, for example, 95% nitrogen, 5% CO2 can be pumped through the system. Now, it's important to keep in mind that if you want to do a study like that on the open bench, that you need to degas the media as well, as most of the oxygen is going to be in the media. There's only going to be a fraction of the oxygen actually coming out of the gas being perfused through the system. The plates are sterilized with ethylene oxide, and if you open them up and you ever look at them, they have a gas-permeable membrane on them. So you can take these plates, put them in a hypoxic box overnight, and the oxygen will go out of the plastic overnight, and you'll have a completely anaerobic plate at that point. We have people that have actually cultured very sensitive microbes where they've stuck the entire microscope and BioFlux into the anaerobic chamber. And that works remarkably well. It keeps the microscope really clean too. But for organisms that are especially sensitive, that are obligate, and any amount of oxygen would be toxic to it, like C. diff, for example. That is definitely an option. And then I have the next question here about, so the question was, they like to see the effect of biofilm on dental material. And, you know, I'm really glad you asked this question, because this is brought up all the time in Dr. Ricard's research. Why aren't you working on porcelain, for example, or other materials that would be used? And the counter argument to that, which I really like and makes all the sense in the world, is that biofilms don't grow on porcelain. They grow on the surface matrix of the polymers from the saliva. And so by coating the channels in the saliva, which coats the proteins and the polysaccharides, that's, you know, in his mind and many other people's minds, just as good as coating in porcelain, except you can see through it. When you think about it, I don't know that any teeth or other dental appliances operate independent of saliva. And I think that's all.
Hey, Brian.
Do you have anything to add?
The last question had to do with the time of biofilm growth. So I think you answered that earlier, but we can go for, you know, as long as 21 days at low shear, and then you know, with the 6-well plates, we can easily do 7 days at a reasonable shear regime. And yeah. So with that, are you ready to close, Brian?
Yes, I think that's the end of the question. Thank you all for attending.
All right. Thanks, everyone. Have a good rest of your day. Thank you.